The study I reported in AACR on behalf of my co-investigator, it's a multi-institutional study of a BCMA CD3 bispecific antibody called Linvoseltamab. Linvoseltamab is a unique bispecific antibody. It is a fully human antibody that binds to myeloma cells by its BCMA binding domain and to the patient's own T cells by the CD3 binding domain. We know that the bispecific antibodies are quite effective. And here I report the effectiveness of the linvoseltamab. This was a phase 1/phase 2 study of linvoseltamab at a dose of 200 milligram. And this was the data that has been submitted to the FDA. So the data cutoff was the submission to the FDA for BLA application. There were about 117 patients who were enrolled on the study. The study design is unique. This allowed for step-up dosing at 5 milligram on day 1 and 25 milligram on day 8, and then 200 milligram weekly. So there are 2 step-up dosing at 5 milligram and at 25 milligram. The patients stay in the hospital just for 24 hours. So there are only 2 24-hour hospitalization involved during the step-up process. Afterwards, the patients are on 200 milligram for the first 14 weeks. And after that, it is reduced to every other week for 24 weeks. And finally, at 24 weeks, if the patient has achieved VGPR, then these patients are transitioned to once every 4 weeks. So the frequency of administration has been reduced at multiple times, such that by the end of 6 months, the patients are, most of the patients are receiving only once a month. The results are very, very encouraging. The overall response rate, as defined by the independent review committee, the overall response rate was 71%, and the VGPR was about 62%, and the overall complete response rate was 46%. So only of the 71% of the patients who achieved overall response rate, only 9% were in partial remission. The rest were VGPR or better, and 46, almost half of the patients were in complete remission. Moreover, when we looked for the MRD status in those it was evaluable, there were 27 patients in whom MRD could be evaluated. 25 of them were in MRD negative at 10 to the minus 5. So it shows that the patient achieved deep response with this drug. So we do know currently that there are 2 other BCMA bispecific antibodies and that are approved. One is teclistamab, and the other one is elranatamab. And this drug is, first of all, is fully human as opposed to humanized with the other antibodies. Therefore, the anti-drug antibody, is very less and less than 1%. And also, infusion-related reactions are very uncommon with this drug. But what is more unique is the overall response rate is very good, 71%, and also the VGPR rate is very high. As I said, only 9% were in partial remission, so 62% had achieved VGPR or better, and also 46% had achieved a complete remission. So this also shows that this is a very effective bispecific antibody. That's another thing. Then the study population in whom it was conducted, the median age was 70, which is what is the normal median age of myeloma in this country. And second is 17% of them were African Americans in the study. And more than a quarter of the patients were over the age of 75. So there is an external validity which the FDA looks for. That means the patients who were treated, the population of patients who were treated, basically reflected the myeloma population at large. So what we found in the clinical trial should be applicable for the wider population. So that is another important aspect of it. And what was impressive is almost 100% of the patients who were enrolled in the study were triple class exposed, and over 85% of the patients were triple class, over 80% of the patients were triple class refractory. This is even higher than the ida-cell patients who participated in Karma 3, which we got recently approved for earlier lines of therapy. So these patient populations were more heavily pretreated, and the responses and the depth of response was phenomenal. And we look at the median duration of response or progression-free survival. The follow-up was short at 11.1 months for the study, but the medians were not reached at all. And so this was very, very impressive, uh, what was accomplished with this, what could be accomplished with this molecule. The next step is, of course, to have a phase 3 trial. There is a phase 3 trial planned for this study, which is the linker MM3 study. And hopefully, this drug will get the nod from the FDA and EMA for the accelerated approval. And we hope that the phase 3 trial will support that application and early approval. And then we expect that the phase 3 trial will be positive. I'm fairly confident because, as I said, this patient population, which was recently approved for the ida-cell in earlier line, more than 2 lines of therapy, the CAR T-cell approval, this drug is able to match the results, what was obtained with the CAR T. So I'm fairly confident it will have a very positive response on the phase 3 trial, and this drug will be immensely of great value for patients with multiple myeloma.